So now that we've talked about enzymes in general terms, let's look at just one specific example of an enzyme and go through its enzymatic mechanism. Of course, we could do this for any number of thousands of different enzymes. In fact, your book actually shows 3 different mechanisms, but I'm just going to go through one of them. And this is a pretty typical enzyme. So this is the mechanism for an enzyme called chymotrypsin. And chymotrypsin is an example of an enzyme whose job it is to cut peptide bonds of other proteins. And so there are lots of different examples of these. So this is an example of a protease, or sometimes called a peptidase, and that means that it breaks peptide bonds. And most proteases are specific for either particular combinations of amino acids or groups of amino acids. So chymotrypsin works by cutting the peptide bond between an aromatic amino acid and some other amino acid. So and it cuts it on the carboxy side of the aromatic amino acid. So it's a little bit unusual actually in that the substrate in this case can be almost any protein, as long as it has an aromatic amino acid somewhere in it. So it's not as specific as some enzymes are. But we're going to kind of just go through and talk about its structure and how it actually carries out this reaction. So the active site for chymotrypsin consists of this hydrophobic pocket, which is going to be the spot where the aromatic side chain is going to stick. So because it's hydrophobic, the aromatic groups side chains are mostly hydrophobic also. They fit right nicely into that little pocket. And then there's this red region called the oxyanion hole, and that's going to be important for stabilizing one of the intermediates. And so here you can see this just the 3-dimensional structure of the protein. The shape of the protein with the active site visible. And then this is just kind of the overall tertiary structure for the protein. And then here's the active site. Again, there's that hydrophobic pocket, and you can see how a peptide fits in there. So the peptide fits in this pocket with the aromatic residue stuck into that little hole there. So this reaction starts, of course, like all enzymatic reactions, with the substrate binding to the active site. So here's kind of a cartoon of what the active site looks like. Again, here's the hydrophobic pocket here, and then that oxyanion hole right next to it. And these residues, which are part of the chymotrypsin protein, are going to be important for the mechanism. So in this case, here's our substrate, so it can be any polypeptide with an aromatic amino acid residue. So here's the aromatic amino acid residue, and it's just it's called R2 because it's the it consists of, in this case, at least 3 amino acids, which are going to actually fit inside the active site. But again, it could be part of a much longer polypeptide. That's why that's what this means. So when we say AAN at this end and AAN at this end, it means there's more amino acids at both ends. And we're just concerned with this one because it happens to be in the active site. And again, this little R2 could be any of the 3 aromatic amino acid residues. And it fits in this way with the again, that aromatic amino acid residue here, the carboxy terminal of the amino acid facing this way, and the amino terminal facing this way, and then the carbonyl oxygen of that aromatic amino acid fits right there into that anion hole, and it gets stabilized again by those other amino acids surrounding it. And so the first thing that happens as part of the reaction is a nucleophile attack. And what that means is these 2 residues, this histidine and this serine, are close enough together that it allows the hydroxyl group of that serine to attack the carbonyl carbon of our peptide that we're cutting, and that means that the pair of electrons with the carbon and the oxygen go to the oxygen, forming that oxyanion. But because it's surrounded by this serine residue and then a nearby glycine residue, it stabilizes that negative charge, And so you have this kind of temporary acylated group forming, but then that leads to the actual cleavage of the bond. So the peptide bond itself that we're breaking is right here. And now that everything's stabilized, that bond, those electrons actually can then attack this extra hydrogen that got picked up by this histidine and take it with it. And that once that bond is cleaved, the first amino terminal of our peptide now is free. So that's the first product of our reaction. And now we have the aromatic amino acid covalently bound to that serine residue. And then, now of course, but we're not done yet because we need to get rid of the other end of the peptide. Now, though, there's space for water to come in. Water then can act as a nucleophile and attack that carbon and sort of repeat the reaction, but now instead of again the amino terminal or that peptide bond breaking, now we're forming a bond with water. And again, that histidine residue is important again because the histidine is going to act basically like a base, and it's going to strip a proton off of that water to make it into a hydroxide ion. And that hydroxide ion then will have the ability to be highly nucleophilic and go after that carbon, which again makes this carbonyl oxygen back into an oxyanion, and then that eventually collapses back into a double bond, but then the bond between the carboxy carbon and that serine residue break down, which then allows that serine to take its oxygen back from that histidine. And so that then allows or separates all the covalent bonds between the peptide and the enzyme itself. And so now you have finally the end product, or the second end product, which is the peptide now with a free or with the aromatic amino acid at the carboxy terminal. And now, of course, the enzyme active site is free, and the enzyme is back to the way it started. And again, that is the important thing about enzymes, by definition, or catalysts in general, is they should end up in the reaction exactly the same way they started out. And now, of course, also now this enzyme is free to break down another peptide if one comes along. So this is again a common example of an enzymatic mechanism. This particular enzyme is found in digestive juices, so it's important for breaking down proteins in your digestive system that are part of your food. But there are other peptidases like this that are also important. So, and of course, this is also a hydrolysis reaction. So it also uses a molecule of water. So again, what's important to understand is that this is typical of an enzyme where you have multiple steps in between, and the final product is what comes out. But in the meantime, you form lots of little intermediate stages that only exist for maybe a fraction of a second, and then almost immediately convert into a different form. So this whole reaction may only take a tiny fraction of a second overall, but there's lots of intermediate steps in between, and one of those steps presumably is the rate limiting step. So it's the step that takes the longest, and is therefore the one that determines the whatever the rate constant is for this particular reaction. Again, the shape of this active site is what really determines two things. It depends the specificity of the enzyme. So the reason that it only cuts peptide bonds next to aromatic amino acids is because of that little hydrophobic pocket. If you had an amino acid or a peptide that was not aromatic, so let's say it was charged or polar, then it would probably have water molecule on it, and it would not be stable in that pocket. Also, the position of these other residues, like this histidine and serine and glycine residues, they're crucial for stabilizing all those intermediate forms. So again, any mutation in this enzyme in the protein that may affect one of these amino residues, even if it didn't change this particular residue. So like this histidine, again, is very important. So if you change that to another amino acid, obviously it would prevent this enzyme from functioning. But any amino acid change or any mutation that would cause maybe this histidine residue to be slightly out of place, again, just a few angstroms out of place, and it might be enough to, if not completely disable this enzyme, seriously alter the binding energy, and therefore also would affect the overall rate of the reaction. Now, the last thing to kind of say about enzymes for now is that enzymes obviously are very powerful. They increase the rate of these metabolic reactions many fold, and for the most part, that's good. That's what they're used for. But the overall metabolic balance in the cell depends on these enzymes working in concert with each other. So you can have enzymes that become overactive or can produce too much of their product or break down too much of their substrate. And so most enzymes have to be regulated in some way. That means that their activity can be increased or decreased depending on various factors, which we'll talk about. So these are called regulatory enzymes, which means that they have some sort of mechanism of being modulated. Now, this actually is pretty much all enzymes are regulatory enzymes in some way. They're affected by their activity is affected by some external factor. So we're just going to talk about some examples here. A common mechanism for regulating enzymes is through allosteric modification. That means that something other than the substrate binds to the enzyme and changes its shape in some way to either usually increase the activity of the enzyme, particularly, and by definition, this would be a positive modulator. So this is just a generic example of allosteric modification here. So we have an enzyme with two domains. You have what's called the catalytic domain, that's what the C stands for, and then the regulatory domain. So this is two completely different parts of the protein. And the point is that the catalytic domain contains the substrate binding site, the active site of the enzyme, and then the regulatory domain contains the binding site for this positive modulator, whatever it is. It's some other molecule, could be a cofactor or coenzyme, but it's just something that needs to bind to that regulatory domain in order to increase the activity of the enzyme. Now, this doesn't mean that the modulator is absolutely necessary, so you could still have binding between the substrate and the catalytic domain without the modulator, but the modulator causes the protein to change shape and basically to either increase the affinity of the substrate or just increase the ability of the enzyme to carry out the reaction. But the presence of that modulator, again, will just increase that activity, and vice versa. So in fact, the substrate also can increase the binding sometimes of the modulator. Sometimes the modulator itself can be a substrate for the enzyme, but other times it can be just a molecule that binds temporarily to the enzyme. Now, the point here is the modulator then can act as a signal, essentially, to sort of turn on the enzyme. So the modulator perhaps could be a product of some other enzyme. And so the enzyme, the first enzyme that produces the modulator, could then sort of turn on the second enzyme by producing the modulator and increasing its activity. Again, it's just a way of regulating enzymatic activity. And another way that this can, so a positive modulator, of course, would increase the activity of an enzyme. You can also have negative modulators. These are substances that decrease the effectiveness of the enzyme when they bind to that regulatory domain. And these also would act as inhibitors. Again, if they're not binding to the active site, we would call them uncompetitive or mixed inhibitors. But in any case, they reduce the activity of the enzyme. This is common when you have what we call feedback inhibition. So sometimes when an enzyme produces a product, either the product itself or something that is made somewhere downstream in its pathway can act as a negative modulator. So this is an example. This is an enzyme, and I forgot what the enzyme is, but it produces a product that then can eventually be modified or converted after several steps into molecules of CTP. So CTP is cytosine triphosphate. And it turns out that this enzyme, even though it doesn't make CTP, has and doesn't react with CTP, has binding sites for it. So CTP actually is a negative. Is a negative modulator. So this enzyme it produces several other downstream products, and then one of those downstream products eventually gets converted into CTP. And in this case, what happens is binding of CTP converts, again, has an allosteric effect on the enzyme to convert it into what's called its active R state, inactive T state. But this is just a generic example of when a negative modulator is bound to an enzyme. It changes the shape, changes the binding site shape or the active site shape, and reduces its activity. But the reason you would want the product of an enzyme or something downstream from the enzyme to inhibit the enzyme is basically like a feedback mechanism. So, in fact, sometimes this is called feedback inhibition, and it's just a way for the enzyme to sort of regulate itself, or for the cell or the reaction to regulate itself. So this is a way for the cell to tell this enzyme that it's got enough of this stuff. So it's got enough CTP. Therefore, this enzyme doesn't need to keep working. It doesn't need to keep making its product. So it's a way of shutting itself down, essentially. It's sort of almost self-regulating enzyme. And then the effect of those allosteric modifiers can also change the kinetics of these enzymes. So this is, again, just looking at substrate concentration versus V0. And here we're looking at the effect of the Km. Here they're calling it the K0.5, but it means the same thing. Remember, that's the concentration, substrate concentration when you're halfway to Vmax. That's what it means by K1/2 here. And looking at the effects of either positive or negative modulators. And in this case, the positive modulator reduces the K1/2, and the negative modulator increases it. So again, that makes sense because the lower the Km, the less substrate you need to get to Vmax, whereas at higher Kms, you need more substrate to get to that Vmax. And you may notice that this curve has that same kind of sigmoidal shape that we saw when we looked at hemoglobin. So again, hemoglobin binding affinity for oxygen has a curve that looks like this compared to oxygen concentration, because again, at low oxygen concentrations, it's in its less active site, and at higher concentrations, it's in its more active state or higher affinity state. And for the same reason, because again, oxygen is a modulator for hemoglobin. Even though hemoglobin is not an enzyme, the effect of that modulator looks the same when you kind of graph it out this way. And then another way to modulate enzymes is through covalent modification. So actually either adding functional groups covalently or taking them away from the protein can usually affect the function of the enzyme. This is a much more long-term mechanism for regulating enzymes than using allosteric modulators. A common way, which we'll talk about more in a second, is phosphorylation, so adding a phosphate group to an enzyme at a specific location. Adenylation, acetylation, these are all different functional groups that can be added to the enzyme at specific amino acids. Usually these groups, when they're added, will obviously change the shape. So if you add a big negatively charged phosphate group, for example, or a big bulky ADP or adenyl or AMP group to an amino acid, that's going to have a big impact on the overall shape of the protein. That'll change the shape of the active site. And so sometimes these modifications can increase activity, sometimes they decrease activity. It just depends on where the modification occurs. Just as a more specific example, phosphorylation is probably one of the most common mechanisms for regulating not just enzymes, but other kinds of proteins as well. So some receptors and ion channels are also regulated by phosphorylation. So what phosphorylation does, again, is it adds a phosphate group to specific residues on the amino acid or on the protein. So you can have, and there are specific enzymes that add phosphate groups to other enzymes, and those enzymes are called kinases. So a kinase is an enzyme which adds phosphate groups to specific amino acids on other proteins, and sometimes those themselves can be enzymes. So this enzyme, phosphorylase, has two forms. So phosphorylase B and phosphorylase A. And phosphorylase B is the unphosphorylated form. Phosphorylase A is the phosphorylated form. Now, phosphorylase itself is not an enzyme that adds phosphate groups. It just happens to involve phosphate groups itself. So phosphorylase's job is to phosphorylate a different molecule called glucagon, but it itself gets phosphorylated by an enzyme called phosphorylase kinase. So phosphorylase kinase phosphorylates phosphorylase, which I know is confusing, but that's just the way these things are named. And so again, phosphorylase kinase specifically phosphorylates certain amino acids on the phosphorylase enzyme. And that phosphorylation changes the shape of the enzyme and changes its activity level. Also, phosphorylation usually requires ATP, so two ATPs are needed to add phosphates to two residues. And then you can remove phosphates with a different class of enzymes called phosphatases. So kinases add phosphates, phosphatases remove phosphates. And in this case, this particular enzyme is dephosphorylated by an enzyme called phosphoprotein phosphatase. So this is, again, removing two phosphates in the form of inorganic phosphates. And this is also a hydrolysis reaction, so it requires water. So you have two different sort of switches here, one which turns phosphorylase on, one of which turns it off. So that's a nice use of these covalent modifications, is they can be added or removed whenever it's necessary to increase or decrease the activity of an enzyme. And again, there are lots of different types of kinases, and most of them are specific for certain sequences or certain amino acids. So these here, all of these are called serine-threonine kinases because they specifically bind to serine or threonine residues along proteins, and they also look for specific, what we call consensus sequences. So particular sequences of amino acids within a protein. So protein kinase A, for example, looks for this consensus sequence. It looks for either a serine or threonine situated next to, can be next to any amino acid, but then next to that you have to have an arginine or a lysine, and then next to that another arginine, and then next to that just any generic amino acid. And then on the other side, this B just means it can be any basic, or basically what that means, in this case, hydrophobic amino acid. And that's its consensus sequence. So protein kinase A will only specifically bind to enzymes or proteins that have that particular sequence. So again, protein kinase A itself is an enzyme, and it can also phosphorylate other enzymes as well as other non-enzyme proteins that just happen to have that sequence. And so each of these kinases has its own particular sequence it looks for. Another group particularly binds to tyrosine, and so they're called tyrosine kinases. So all of these insulin receptor kinase, EGF receptor kinases, they specifically look for tyrosine residues within these consensus sequences. So each one, again, looks for a particular region. So they're not just phosphorylating any enzyme. They look for particular, or any protein, they have to look for particular consensus sequences, and only those proteins that have that sequence get phosphorylated. Again, here what you're seeing is the phosphorylation sites on one particular enzyme. So this is the enzyme glycogen synthase, and it has at least 1, 2, 3, 4, 5, 6, 7, 8, 9 different sites that have the ability to be phosphorylated, and some of them can be phosphorylated by the same enzyme. So, for example, site 1A, this one here, can be phosphorylated by protein kinase A, protein kinase G, or protein kinase C. And then protein kinase A, for example, can phosphorylate at least 4 different locations on this particular enzyme. And then there are others like casein kinase, which has the ability to phosphorylate up to 9 different locations on this particular protein. And different phosphorylation sites have different effects on the protein. So over here you see degree of synthase inactivation. So all these sites, when phosphorylated, reduce the activity of the enzyme, and some of them more than others. So again, casein kinase 1 has a much greater effect than all the rest of them, but then a very specific one called glycogen synthase kinase has a pretty large effect too. So there's no way to predict just from the structure of a protein whether phosphorylation will increase or decrease, or the activity of an enzyme, or how much it will increase. And so the effect of phosphorylation is part of this kind of very complicated network of phosphorylation and dephosphorylation signals. Another important mechanism for regulating enzymes, or feature of some regulatory enzymes, is that they are actually activated when when they're cleaved from a larger precursor protein. So chymotrypsin, which we talked about earlier, goes through kind of 3 stages. So the final form of chymotrypsin is the cleaved form. So the active form of chymotrypsin, which is called alpha chymotrypsin, is actually 3 separate polypeptides, but they start off as a single peptide. So when chymotrypsin is synthesized, when it's made or translated by a ribosome, it's this long 245 amino acid protein, which is called chymotrypsinogen. And in fact, the generic name for one of these proteins that starts off as a longer enzyme and then gets broken down, or a longer protein that gets broken down into its active form, is called zymogens. So this is the zymogen for chymotrypsin. So chymotrypsinogen is just its long name. And it first gets cleaved by a different peptidase called trypsin. So trypsin cuts this protein at the 15th amino acid, so between amino acids 15 and 16. And again, it looks for a particular consensus sequence. So it looks for this arginine isoleucine boundary to cut. And so that form of chymotrypsin is called pi chymotrypsin, and it also is active, but it becomes more active when it gets cleaved again at this boundary between this tyrosine and this alanine. So that's amino acid 146 and 149, and it turns out that this particular cleavage is carried out by pi chymotrypsin itself. So this is called autolysis. It just cuts itself into again from 2 pieces into 3. And in so doing, it actually removes a couple of residues. So it loses numbers 147, 148, and then it also loses 2 more from amino acids 14 and 15. But this then is the final primary structure of this enzyme. So it's 3 subunits that combine together. They're attached by disulfide bridges, and this forms the final version of the enzyme. And so this is a way of regulating enzymes. Another one is trypsin itself. So again, trypsin makes the first cut from the zymogen for chymotrypsin But trypsin itself also comes from a zymogen called trypsinogen, and it gets activated by a different enzyme called enteropeptidase. And enteropeptidase just cuts off the first 6 amino acids of trypsinogen to form the active form of trypsin. So this is another way of regulating enzymes. You can have these zymogens hanging out in the cell, inactive, and waiting to be turned on or activated by cleavage. So when chymotrypsin is necessary or when it's needed, trypsin can come along and cleave out, start the cleavage process that actually activates the enzyme. So what's important to understand is that at any given time, an enzyme, it may be present in the cell, but its activity is regulated by lots of different factors. So here's again our enzyme phosphorylase B. Again, phosphorylase its job is to phosphorylate glucose or start the process of breaking down glycogen or glucagon into glucose. So it has lots of different regulatory signals, and some of those. So again, phosphorylation itself, phosphorylation by phosphorylase kinase regulates that enzyme, but then there are also lots of modulators that can affect it. So we'll learn later that phosphorylation of glucose is an important step in breaking down glucose for energy and eventually producing ATP. So ATP itself actually is a negative modulator for this enzyme. Again, that's another example of sort of feedback inhibition. And then AMP, which is sort of the version of that when ATP is broken down, it can eventually form into AMP. So high levels of AMP would be a signal to the cell that it's low on energy. So AMP acts as a positive modulator for this enzyme. And again, the phosphorylation and dephosphorylation process itself is also regulated. So phosphorylase kinase, again, its job is to phosphorylate phosphorylase B into phosphorylase A, but it also gets phosphorylated by protein kinase A. So protein kinase A can phosphorylate phosphorylase kinase and turn it from a less active to a more active form. So that's what's happening here. And then another phosphatase, actually the same one that dephosphorylates phosphorylase, also dephosphorylates phosphorylase kinase. Again, I know this is all getting confusing. We're not going to memorize all these pathways. I just want you to appreciate kind of how complicated this enzyme regulation system can be, because those, again, the thing that can modulate phosphorylase itself can be modulated by phosphorylation, and those factors can also be modulated further. So protein kinase A phosphorylates phosphorylase kinase, but it also is regulated or can be positively regulated by cyclic AMP, which in turn can increase in the presence of glucagon, which is one of the things that phosphorylase works on. It's the substrate for phosphorylase. And meanwhile, protein phosphatase 1, which is the enzyme that removes phosphates from phosphorylase, can also be regulated. So it also can be phosphorylated by protein kinase A, and it can be dephosphorylated by a different phosphatase. And each of those also have their own regulatory mechanisms. So, and this is not just for regulation within the cell, it can be useful for regulation and communication between cells. So protein phosphatase 1, for example, can be positively modulated by insulin. Insulin is a hormone that's released by your cells when when sugar or glucose levels in your blood are high. And so one of the ways insulin works is by affecting this pathway as well. So again, every enzyme, usually when we study enzymes in the lab, we're looking at just one enzyme in a test tube with its substrate or maybe an inhibitor or a modulator. But it's important to understand that every enzyme, in fact, every protein is working in your cells in concert with a bunch of other proteins and a bunch of other positive and negative modulators. And so the interaction between these proteins can become pretty complicated pretty quickly. But that's as far as we will go today with enzymes. And then after this chapter, we will finally be ready to move on to other kinds of molecules.